To organizer for giving me opportunity to present my work. My research topic is simplified rapid membrane-based viral DNA isolation method for molecular diagnostic of graft transmissible pathogen in citrus. Citrus is an important crop in India. Here are so many variety available like sweet orange, acid lime, rankul lime, pomelo, rough lemon, mandarin, and grapefruit. Citrus are Rutaceae family, tribe Citri and subtribe Citrini, genus Citrus. In India, area is 0.66 million hectare. Production in India 4.78 million ton. This is a major growing area of citrus in India. So many diseases involved in citrus. Important disease of citrus: citrus dystonia, Indian citrus green ring spot, citrus mosaic virus, citrus greening, citrus exocotis, citrus gamoxis, and citrus canker. Citrus mosaic virus are important disease in India. This is a Banda virus group. First reported in India, Dakshamurthy and Reddy, 1975. And disease incidence 10 to 70% reported by Allahabad et al. And 77% reduction in fruit yield, 10% reduction in juice and ascorbic acid. In citrus mosaic Banda virus, Allahabad et al. 1996 did partial characterization of citrus mosaic virus, which they name as citrus yellow mosaic virus. Family Columbidae, genus Banda virus, non-blue bacillifera virus, 30 into 120, 150 nm diameter circular DNA, and then full length genome is 7559 and have 6 overlap. So this is a visual symptoms of citrus mosaic Banda virus. This is a virus electron micrograph showing bacillifera particle. Then another disease are also important in all over world, citrus greening. Citrus greening disease is also important in India and other kingdom and orange area. The citrus greening bacterium catching greening disease, also known as hanglobin. Name of the pathogen: Candidus levibacter asiatus for Asian greening bacterium and Candidus levibacter africans for African greening bacterium. The citrus greening disease is an important disease of citrus and has been reported in different citrus species in several parts of India. It can crop losses up to 30 to 100%, and citrus production may be eliminated in some citrus production area, Tagasa 1991. So this is a visual symptoms in Kindo mandarin orchard, symptom induced by citrus greening on Kindo mandarin under field condition. Or this is another slide is orange fruit affected by citrus greening bacterium. So in best study, my object is simplified rapid membrane based viral DNA isolation method for molecular diagnosis of graft transmissible pathogen in citrus by PCR. Citrus is an important fruit in India and grown in about 0.49 million hectare with an annual production of 4.39 million tons. Citrus mosaic is widely distributed disease in India, especially sweet orange, pomelo, rankul lime. It is caused by bacillifera non-pueblo DNA virus group under the genus Banda virus of family Columbidae. Virus particle measure 130 nm and contains sulfur double stranded DNA. So therefore, content detection by slew method is not a preferred one. So therefore, possibility were exploited to detect the virus by polymerase chain reaction in different tissue in infected citrus plants. So material methods, the culture of CMV were maintained on sweet orange, pomelo and rankul lime in the glasshouse condition. The disease produced mild to high yellow molting symptoms in different species of citrus. In order to determine the distribution of citrus mosaic virus in the infected citrus plant, PCR detection was used to determine the presence and relative distribution in leaf, bark, and bud tissue of infected sweet orange. So pomelo and rankul lime, the DNA template was prepared by simplified NCM-based protocol and commercial kit and was used to standardize by PCR reaction. So then PCR polymerase chain reaction. Then we have taken according to master mix to use for PCR detection. So this is a simplified NCM-based DNA template preparation method for PCR. So we will take 100 microgram of leaf tissue from CMV infected sweet orange plant and 100 gram of petiole and midrib of leaf from candidate L-acetyl, because the concentration of mosaic virus in sweet orange in leaf tissue or bark and other is जो citrus greening are also midrib of leaf, which good content compared to leaf. Sample were ground in 1 ml of alkaline solution, 15 mm NaOH and 2.5 mm EDTA using a slight pestle and mortar. No liquid nitrogen was used for grinding of tissue. So this is a layman technology. The resulting extract was incubation at room temperature. Then 5 ml of sap were 60 spotted on untreated nitrocellular membrane, NCM VS 85 pore size 0.4 by mm. The spotted membrane was dried for 30 minutes. Then individual spot 4.0 mm for each sample were cut out using a paper hole punch kangaroo. Then blood was eluted in 30 ml of external distilled water by incubating 80 degrees centigrade for 10 minutes. The liquid was collected by centrifugation, volume of 2.5, 10 and 20 ml, used for PCR for citrus greening bacterium, citrus mosaic banda virus. So this is a picture of just take sample, then mix slowly, mix extraction, then blotting, then use template for PCR. So this is a nitrocellular membrane blot on the blot on nitrocellular membrane, which is isolated from NaOH EDTA, extract of CMBV and citrus greening bacteria. So this is a electrophorese detection of CMBV and citrus greening bacterium using direct alkaline safe and NCM eluted membrane. So this DNA was extraction by NCM membrane and then used for direct PCR. So the bearing volume are also affected in this technology. So effect of bearing volume of nitrocellular membrane eluted extract for PCR. So first is 1 to 3 line, each take 20 microliter, then 4 to 6, 15, then 7 to 9, 10, and 10 to 11, 12 taken 5 microliter. Then comparative to analysis for which concentration is good for PCR. So another photo is a figure 0.6 comparison of NC eluted extract comparison. This NCM membrane based DNA isolation compared to other method like kit method, Poison kit, Germany, and then sodium sulfide method. So first 3, 1, 2, 3 by eluted by NCM membrane, you see clear cut amplification of citrus mosaic virus with 537 base pair band observed, and also 13 number is marker. And then 5 to 7 isolated DNA by Poison Germany kit, then 9 to 11 by sodium sulfide. So first picture is citrus mosaic banda virus. Next picture is citrus greening bacterium. So this is a good technology for large scale of survey and collection of sample where is not available other facility. The real-time discussion. Amplification of citrus mosaic virus was observed in bud, leaf, and bark of all the 3 citrus varieties used in the study. However, the intensity of PCR product was maximum in bark using DNA template obtained either by membrane or commercial kit. The study indicated that NCM-based DNA extraction would be useful for PCR detection of CMBV in bud, bark, and leaves. So these studies indicate that the bark can be good testing material for detection of citrus mosaic virus, and further dilution studies show that the concentration of citrus mosaic virus was better in bark compared to leaf. PCR application of citrus mosaic virus was observed when DNA isolation by simplified membrane-based method or by commercial kit. So the intensity of PCR product was slightly more than DNA from commercial kit was used. It is concluded that bark can be better source material for PCR detection of CMV, indicate citrus plants. Nitrocellular membrane-based DNA isolation method, an alternative to commercial kit, as reported by Singh et al. 2006. So conclusion, NCM-based DNA exchange protocol is simple, inexpensive, rapid, and applicable to large-scale survey of citrus tree for nursery purpose and other healthy plant material purpose. Then reference Singh et al., Dwarf, A.D. Singh, and M. Clark, 2004. Invention of simple membrane-based nucleic acid preparation protocol for RT-PCR detection of portovirus from aphid and plant. So this technology we will also use for another RNA or DNA virus for DNA extraction purpose. Thank you. Thank you for organizer Chicago, USA and Magnus Group for giving me opportunity for oral presentation. Thank you very much.